Hi guys, and welcome to my screencast. My name is Emma Weber, and today our two main ideas are going to be neuron signals and synaptic transmission. We're going to begin our screencast by reviewing a little bit about the basic neuron, so that way when we go through all the different processes, we know exactly what we're referring to. Then we're going to talk specifically about synaptic transmission, then a little bit about neuron signals, including sodium-potassium pumps and how they build up action potential. And we're going to end by going through a few real-life examples. Okay, so let's start by talking about the structure of a typical neuron. The typical neuron is split into two parts. We have the cell body on one side, and the rest is considered an axon. The cell body is the factory of the neuron, as it contains the nucleus. Meanwhile, the axon is responsible for conveying electrical signals. These guys hanging out over here are known as the dendrites. They technically are on both this side and this side. But let's talk about the ones on the side of the cell body. These tree-like structures are responsible for receiving electrical signals from other cells. Meanwhile, the ones at the end, where we have our word labeled axon terminal, are also dendrites, but these ones are actually known more for the axon terminal. The axon terminal is where the synapse is located, which is responsible for sending synaptic transmission signals or electrical signals to dendrites on other cells. It's important to recognize that this cell will be sending signals from this end, these guys over here, to these dendrites on whatever cell it's transmitting to. Also on the axon, we have a few other important parts. We have the myelin sheath, which are these big yellow dots. And within the myelin sheath, we have Schwann cells. The nodes of Ranvier separate each myelin sheath. It's important to note that electrical signals jump over the myelin sheath from node to Ranvier to node to Ranvier. They don't go through the myelin sheath like a tunnel. This is called saltatory conduction. When a nerve impulse reaches the synapse triggers neurons to release chemical neurotransmitters. These neurotransmitters are located in their synaptic vessels, right here and here, and they go down and get released from the vesicles and travel across the synaptic cleft to the target neuron. Then they fit into receptors on the dendrites of the target neuron, which grab the neurotransmitters and they fit perfectly right in. Then the receptor, neurotransmitter in the receptor continues on through the dendrite and throughout the receiving neuron as a chemical signal. But a big question may be, where does the synapse get the energy for this nerve impulse that sends the synaptic transmission? The answer is a theory called action potential. Action potential is known as the change in membrane voltage that allows for signals to be sent in a neuron. Currents indicate the flow of positive and negatively charged ions across the resistance of the cell membrane, as shown in this photo. So a resting neuron has, for the majority, has the negative potassium ions on the inside of the membrane and the positive sodium ions on the outside of the membrane. This is known as the resting membrane potential, and this resting membrane potential tends to be at negative 70. So we have a few different types of things that allow for there to be change in the potential, allow the potassium and the sodium to move across the membrane. The first is the sodium-potassium pump. So for every 2 potassium positive ions that the sodium potassium pump lets in, it lets out 3 sodium positive ions. This is why there's a more positive charge on the outside and a more negative charge on the inside. This difference is known as electrochemical gradient. And as we've learned throughout our science classes, whenever there's a gradient, especially in the human body, the body works to cancel out that gradient, which is where the pump comes into play, but specifically where ion channels come into play. So ion channels won't have both potassium and sodium moving throughout it. It'll have either one or the other. For example, here we have a potassium pump, and here we have a sodium pump. So in the sodium pump, only sodium will come in, and the potassium pump channel, only potassium will go out. And those, when they open, they can move freely. There's a few different factors that allow them to be opened, but the one that's important for us are the voltage-gated channels. So what's important to understand with this image is that the movement of ions is really the key to electrical events throughout neurons. This movement of ions is what's going to build up the action potential and allow for neuron signals to be sent. Any movement that's big enough to trigger the voltage-gated channels to be open, so anytime there's a big enough difference in or big enough gradient within the inside of the outside of the cell, we're going to reach the threshold, and this will be, the voltage-gated channels will open, and this is known as action potential. We're going to look more at that in our next slide in our graph. So in the process of action potential, we begin in our resting state, which we previously learned was negative 70 megavolts, as shown on the graph right here. So right here we're resting, and then suddenly something happens, something that triggers a neuron, the neurons that have to send a neuron signal. So when this happens, your sodium channels open, and your sodium starts drifting into the neuron. And as we just learned, this is going to make your cell a little bit more positive. As long as it goes up to negative 55 megavolts, you've reached your threshold. If it's a little bit less than that, it's a really small signal, it's not going to work. That's what our failed initiations are. But if it hits negative 55, you're going to hit action potential. It's an all or nothing phenomenon. So you don't hit negative 55, you don't get a signal. You hit negative 55, you have action potential. And suddenly the cell is massively depolarized. The sodium floods into the cell. It goes so far actually to depolarize it that it actually makes it positive. By the time you reach action potential, you're going to be at positive 40 megavolts. It's also important to recognize that this all or nothing phenomenon, it doesn't happen all at once. So it'll send from one will change, negative, positive, negative, positive. As you can see, these are still positive, negative though. So then once these go, then it'll be the next person's turn, the next person, and so on. So it doesn't all flood in at once because that would be a crazy gradient. It happens one at a time and they trigger their neighbors. But back to the graph, once this all or nothing phenomenon happens and it reaches action potential, the neuron's immediate reaction is to try to repolarize. In fact, it tends to repolarize so fast that it actually goes a little bit too far. That's what we call hyperpolarization. So it dips all the way down here before eventually returning to its resting state and doing the neuronic signal all over again. So that's action potential. Now let's try to relate this to a real-life example. A very common and popular painkiller is Novocaine. But how does Novocaine work? Knowing what we've just learned about neuron signals, whenever you feel a pain or something and it goes past a threshold, the nerve should send a signal. However, Novocaine works by blocking the neurons from communicating. The drug works as it blocks the ion gates. Therefore, the neuron can never reach its action potential, can never get past its threshold because there's not enough sodium coming into the neuron. There's not a big enough gradient. Therefore, no signal will ever send. So that's it. Thanks for watching.